Yes, thank you, Professor Kim. It's such an honor to be here, and thanks everyone for joining in. Today I'll be talking about two themes. First, give you a broad overview of single-cell transcribed cis regulatory elements. So we just heard a very nice talk about ataxia, but how we can leverage the enhancer transcription activity using 5' strategy. And second part, I'll also talk about elucidating long non-coding RNA function in subcellular resolution and understanding their role in different compartments within the single cell. So I think we all know now that our human body consists of multiple organs, and according to ChatGPT, there are about 37.2 trillion cells, which are grouped and categorized into a very well specific units as cells that exhibit a specific function. And we know, just like an atom in an element of a periodic table, cells have different genes that can dictate what the function of that cell can be. And they're very interchangeable in many ways. So the question really is, what are these cells made out of? And in order to study that, we need to develop technologies, but also which cells are really important for causing the disease. And this really fundamentally comes down to what is the genetic makeup and the trigger points in the genome that can lead to a pathology from a starting point from a single cell. And this really is the idea that there's a genetic variance in our genome, and in the context of different cell types, that can lead to a different disease. So if you have genetic variance in the neurons, can more likely to lead to autism, whereas in dendritic cells would be more Crohn's disease. So fundamentally, the key importance here is that genetic variance has to be understood in the context of the cellular environment. And so we need to really look at this side by side. And we all know that our genome is extremely complex, and these chromatin arrangements are very cell type specific, and therefore we also need to understand and elucidate their makeup across different cell types in a systematic fashion. So fundamentally, our lab is interested in looking at promoters and enhancer interactions, and we call these cis regulatory elements. And if they are in harmony and they are working as it should, we expect a regulatory network to form to provide a homeostatic, healthy outcome. But once you have these mutations or SNPs in the regulatory regions, such as enhancers or promoters, these interactions can be disrupted, which can lead to unwanted consequences. But going back to this notion that the cellular context is so important, because if you have the SNPs in this enhancer, but this enhancer is not active in endothelial cells, you're actually unperturbed. But if this enhancer is active in the GABAergic neurons, for example, the interaction can be perturbed and could lead to unwanted consequences. So to study this, we really have to understand not only where the SNPs are, but also which cell types these SNPs are most active in. And we leverage this technology called CAGE, or 5' RNA-seq, which allows us to capture the 5' ends of the RNA, which then can be mapped to the genome to understand where the transcriptional start sites are, TSS. And these could be very contextual to which cell type they are in. And many reports have shown that the 5' RNA-seq method is actually extremely powerful to understand promoter activity. And from this paper, notes that DNA-seq or ATAC-seq and H3K4 trimethylation were not efficient or not sufficient without CAGE or 5' information, because really the transcriptional start site readout is a functional readout of the polymerase II activity, which is indicative of the function rather than a passive or paused notion of the genome. So this goes back, way back, actually, many years ago in Phantom Consortium, which started in Japan, weekend, where my former lab was located. And we really profiled many different tissues and cell lines and mapped these promoter atlas. Of course, these were done in bulk. So we wanted to move into single cells. And thankfully, with 10x genomics, we were able to implement this 5' capture strategy just using 10x genomics droplet method. So here's a classical example of what we get when we profile PBMCs. Here in purple, you have this anti-sense direction of NR4A2 gene going right to left, and you have a very strong proximal transcribed cis regulatory elements. And these are overlapping nicely with the chromatin accessibility. Here, upstream of NR4A, interestingly, you have this bidirectional transcription of RNA, which also happens to align very well with open chromatin, but these are not annotated in the gene code. And we now know that these are enhancer RNAs or RNAs that are transcribed from enhancer regulatory elements. And these could be captured from the same single cell using the 10x genomics platform. So it's kind of giving us kind of a two promoter and enhancer activity in the same single cell. And we developed the software that will help us to annotate these transcriptional start sites called SCAFE. And you can see here on the right, the different gene here, transcriptional start sites from right to left. But if you look on the downstream of this gene, you do see these bimodal peaks, which overlaps very nicely with the ataxic peaks in green here. So these are indications of open chromatin regions that are actively transcribed versus there are actually open chromatin regions that are not actively transcribed, and these may indicate passive enhancers. And what's kind of powerful with the transcribed activity is that we can, they're far more dynamic in their gene expression and their activity compared to the ataxic. So the ataxic here, if you stimulate the cells, these are PBMCs stimulated with PMA that activates the T cells. You can see that these resting and stimulated states remain the same, relatively the same, whereas in the expression level, you see a significant dynamic changes in their activity. But what's surprising is that there's an observance of promoter switch, where same gene, but different promoter is activated upon the stimulation. And this can be readily captured by RNA-seq as opposed to the ataxic chromatin. So hopefully this kind of gives you an importance that the switch promoters can be readily observed using RNA-based detection of the promoters compared to ataxic. And because they are really dynamic in their expression, we can you can observe their co-activities. Here's a PTGR4 locus. And upstream of this, we have very large clusters of ataxic peaks, but also we can observe large clusters of enhancer RNAs being transcribed. And we call these regions a super enhancer RNAs, where you have clusters of multiple eRNAs transcribed, and which happens to overlap with the SNPs that are found in Crohn's disease in the non-coding region rather than the coding region of the genome. So using this 5' single-cell RNA-seq technology, we went out and collaborated with various medical institutions in Japan, because RIKEN is not a medical institute. We are a research center, so we needed to collect clinical samples. And we were very fortunate to work with various clinicians to provide us with very valuable patient samples, where we collected about 23 tissues and profiled over 400,000 single cells and mapped these transcribed CRAs as follows. So you can readily appreciate the immune clusters and the epithelial and stromal clusters, as well as neuronal clusters. And of course, the power of single cells is that you can subcluster them to gain higher granularity of subtypes, such as GABAergic neurons have multiple subtypes, as we know from literature. But ultimately, our goal was to integrate our TCRE or cis regulatory element activities of both promoters and enhancers together with all the GWAS genetic traits that have been summarized for the past decade or so, and understand what are the transcriptional activity in the promoters and enhancers co-localized with all the genetic SNPs that have been characterized in the past. So here is another example of, say, lung alveolar cells may have a strong activity which co-align with GWAS SNPs, but these SNPs may not be so relevant in neurons, for example. And that can then help us to associate that this genetic SNPs is more relevant for lung disease versus brain disease. So to systematically develop this tool, we needed to perform multiple build a pipeline. So here is a first single cell clustering, and we perform meta cell clustering. And this meta cell clustering was then transformed to understand their gene programs or modules. And these were then later utilized for cell type clustering. Together, now once they are in the module and cell type format, we ran a linkage disequilibrium regression analysis to then associate these disease SNPs to these modules and found statistically enrichment versus the background. And what that allows us to do is then link the modules of different cellular modules to the heritability enrichment of those SNPs, and then further deconvolute back to individual cell types, and then find these enrichments to individual cells. So that's, in a nutshell, the workflow of this strategy. If you were to visualize this in actual analysis, here's the single cell clusters that I've shown earlier. They're the meta cells. We annotate these meta cells, and then we will perform linkage of disequilibrium to then associate various disease SNPs to the modules and trace it back to the single cell map to say which of the SNPs are then enriched in which cell types. So here's a summary of various, it's actually a snapshot of many other hundreds of different GWAS summary statistics. But here you can appreciate the clusters of different cell types, such as immune cell types, enriched for GWAS SNPs that are associated to eczema or asthma and Crohn's disease, where you have neuronal cell clusters that are enriched for schizophrenia or mental distress, or you have the stromal cells, such as endothelial cells or fibroblasts that are enriched for blood pressure or myocardial infarction, as you would expect. But keep in mind that these are now single cell type resolutions, so we can even, these are what we call level 1 clusters, so we can even further zoom into individual subtypes to see which of them are much more enriched at the subtype classification level. So here's just an example of psoriasis, where you have T cell enriched endothelial cells and dendritic cells, but arthritis have completely different subtype of T cells enriched for this disease, and B cells and plasma cells. As I mentioned, it can go into higher resolution with the venous endothelial cells, capillaries or arterial endothelial cells, and different subtypes are associated to different disease traits, such as varicose vein are enriched in venous osteochondrosis in arterial endothelial cells, and triglyceride in the capillaries. So this is a same representation but different color. But what we can do with this type of data set is we can contrast different traits. So here we can contrast Crohn's disease versus ulcerative colitis, and we can see which immune cells are quite different between these two disease types. So here we see that any stromal cells and perhaps some monocytes are more important for Crohn's disease, where ulcerative colitis are more enriched for T cells, as you would expect. So these are type of really nice modalities that we can start contrasting disease of similar origin and really get into finer resolution of cell types that may be causal to these disease. There's another, it's not a disease, but a trait. Height is really well studied GWAS trait. We do find stromal cell clusters that are highly enriched, as you would expect. We can even look at within the same chondrocyte clusters that there are different varying degrees of enrichment for height. So here on the left versus on the right. And we can rank this, that the genes that are much more enriched in the left-hand side of this chondrocytes are WWB2 gene, where on the right-hand side, or the anti-correlated to height, are SMAD7. What's interesting is that these two genes actually are in the same regulatory network, and that SMAD7 can interact with various pathways to regulate the ossification, which then cause the activation of this chondrocyte growth. Just to note that this SMAD7 is actually regulated by WWP2 gene. So these are quite very interesting extrapolation of understanding the traits to not only the cell types, but also which genes are actually causal and regulating these pathways. So to summarize this part, I hope I've shown you that 5' RNA-seq really allows us to identify cell type specific promoters and enhancers, and that these transcribed CREs are enriched in these GWAS SNPs, and they also reveal these alternative promoters and enhancer usage. And hopefully I've kind of shown you that this TCR-ALX connects the SNPs variants found in promoters to gene programs to specific cell types, where previously we were associating SNPs to disease traits, but now SNPs can be linked to a promoter or enhancer, which can then be linked to gene programs, which are part of a cell type that can lead to a disease trait. So all of this work is actually summarized in this paper, which we just put it up. So feel free to read this, and hopefully we can share them with you in a published format soon. I'm going to switch gears quickly and talk about transcription in the non-coding region, because it's sticking with the theme of non-coding regulatory elements. Our lab was also very interested in studying long non-coding RNAs, partly because, as we know, that these non-coding RNAs are extremely pervasively transcribed across the genome. Here's in blue is protein coding gene, but in red are non-coding RNAs. This can happen in cis, but also in trans and across the multiple regions of the genome. And so we really want to functionally understand their regulatory role. And again, going back to this slide, many non-coding RNAs does play a role in promoting interactions and histone modifications and DNA methylations. And biologically important also in development as well as in disease. So our effort with Phantom also have led us to identify about 27 or 28,000 long non-coding RNAs. And a completely independent group in Europe have discovered a very similar number of non-coding RNAs across multiple tissues. However, many of them are not yet published, and they still need to be functionally characterized. So we do this by taking two different approaches. One is pooled library screening, where we use CRISPR-I to pool multiple guide RNAs, and we do a phenotypic selection. Or we do an array-based screening, where we knock down individual non-coding RNAs using antisense oligos. So I just want to give you a snapshot of what we've been doing with CRISPR-I. We designed multiple guide RNAs targeting about 1,300 non-coding RNAs, and we targeted the iPS cells during the proliferation stages, as well as differentiation. Here's just an example of one non-coding RNA that was highly enriched, link 1816 and link 173. which are significant, what we call depleted, actually, in the cluster in the cells during the iPS proliferation. This is very promising because other protein coding genes like POU5F1, which is an OCT4 gene important for pluripotency, is also depleted. And we were very intrigued by these two non-coding RNAs. We were able to independently validate that these are indeed important in regulating cell growth of iPS cells. And this multi-omics analysis have led us to link these non-coding RNAs to this protein coding gene MXD1 in a cis-regulatory manner through chromatin-chromatin interaction. And once we deplete this non-coding RNA, this MXD1 protein coding gene is actually upregulated. And this is very interesting because MXD1 is a cofactor of MYC, one of the key oncogenes, and it actually suppresses MYC through binding of MXD1. So in summary, the link of RNA suppression upregulates MXD1, binds to MYC, and suppresses the activity of MYC, thus leading to reduction in cell growth. So it's just one example of how we can use pooled screening to identify a non-coding RNA. But we will also have approached this array-based screening using antisense oligos. These are single-stranded DNA that can hybridize to RNA, and that RNA-DNA duplex elicits an RNase H cleavage, then suppresses the gene activity. So we designed about 5 ASOs across multiple non-coding RNAs in iPS and fibroblasts. We used robotics to perform functional genomics screening, and we profiled both cellular phenotype as well as molecular phenotype. Here we perform RNA sequencing after the perturbation of suppression of these non-coding RNAs. So here in the cellular phenotype, we do identify a set of non-coding RNAs that are significantly important in the survival of cells, and that they do lead to significant suppression of cell growth. Interestingly, non-coding RNAs that are highly expressed, or they are cell type specific, or they have high C interactions, tend more likely to lead to a cell growth phenotype than other properties. But interestingly, we also performed RNA sequencing and profiled their gene transcriptional activities upon the depletion of these non-coding RNAs. And some non-coding RNAs lead to very little transcriptional changes, but some non-coding RNAs lead to very significant changes in gene activity. And these differential expressed genes can be then mapped to functional pathways and try to understand then the functional associations of these non-coding RNAs to various classes of GO ontologies. I'm kind of going through rather quickly because all of this work is published, and you can actually go to Zenbu reports and you can survey individual genes that we have mapped now and look at their differential gene expression analysis and their localization and so on. But what was also interesting is that we took one of these non-coding RNAs. We wanted to functionally validate their mechanism. So we did a pull down using CHIRP. And this is a method developed by Howard Chang's lab, where we designed multiple probes against the non-coding RNAs and perform genomic DNA sequencing and RNA binding protein mass spec. And that allowed us to understand that these non-coding RNAs actually acts in trans by interacting with different regions of the genome. And they actually interact with one of the key RNA binding proteins called RBPP7. And if we knock down these non-coding RNA, or if we knock down these protein coding genes, which interacts with this non-coding RNA, can lead to a significant depletion of cell phenotype, cell growth rate. So here's just a summary of this non-coding RNA, which binds to this RNA binding protein, which can then interact with the histone as well as the transcription activity, and can either modulate the KLF4 expression or SEMA3C to change the cell growth, the cell migration patterns. But we were interested in the functional readout, but we were also curious about their subcellular localization of the non-coding RNA. And this is rather interesting because non-coding RNAs play a key role not only in the nucleus, where I've just shown with the previous example, but they can also regulate in the cytoplasm by modulating translation. So although we've been looking a lot with spatial transcriptomics, works have been investigated quite a bit. I think it remains to be seen how this non-coding RNA localization in the subcellular level could also provide very new functional and biological insights in the future. So to study that, we first stimulated a human dermal fibroblast with various drugs that can perturb a key biological processes within the cell. Just to give some examples, actinomycin D can inhibit transcription, where cyclohexamide can inhibit translation, where 5-AZA-D and BPA can modulate the epigenetic programming of the cell, and so on. So we stimulated these drugs and fractionated the cells into cytoplasm, nuclear soluble, which are RNAs that are in the nucleus, but they're not bound to the chromatin, as well as chromatin-bound RNAs. So these three fractions then perform RNA sequencing and allow us to perform this global distribution analysis using this ternary plot, where you have RNA distribution from cytoplasm, chromatin-bound to nuclear soluble. And since we now have two different conditions, we can compare vehicle to drugs and understand how these RNAs are co-localized or displaced within the cell from chromatin fraction to the nuclear soluble, or chromatin fraction to the cytoplasmic fraction. And whether these could be indication of their function was the aim of the study. And we identified one, and we'll highlight some examples in the following slides. So here is just an example of a RNA profile of actinomycin D treatment. As I mentioned, actinomycin D inhibits transcription, and it pauses the transcription in some ways. So you have actual enrichment of RNA that are bound to the chromatin versus the control. And we have other control experiments where CSA can have a condition where chromatin bound can move into the central part of this ternary plot. We have a 5-AZA-D, which inhibits DNA methylation. We have a vehicle in the middle, but some RNAs move to the chromatin, some RNA move to the soluble fraction, and some RNA move to the cytoplasm. BPA, which is a HDAC inhibitor, can move also from chromatin bound more to the cytoplasm or to the nuclear soluble. So we have very interesting dynamic patterns that are observed of RNA localization within the cell across different perturbation. But for the time, I just want to focus on BPA and illustrate to you how these RNAs are translocated. So here is, again, a ternary plot illustrating the RNA localization. Now, if you put the vehicle to the center of this graph, you can actually project the direction in which the RNA is localized from control to the perturbed state. So everything in the middle is a control, and they're moving away from their site of origin. And you can see that many messenger RNAs do move to various locations, but interestingly, non-coding RNA class move more in the nuclear soluble fraction, as observed here. And we were able to validate this using single molecule FISH, and that one of these non-coding RNA actually has a localization into nuclear soluble fraction, moving abundantly across the nucleus upon the stimulation of BPA. Again, BPA is an HDAC inhibitor, which inhibits demethylates acylation marks, and we see an enrichment of this non-coding RNA called MIAT in the nucleus. So we also perform a ChIRP-seq again. So we stimulate the cells with BPA, design the probes against this non-coding RNA MIAT, and we demonstrate that they do cross-interact with multiple chromosomes in the genome. And interestingly, a lot of the interacting partners are enriched for acetylation-dependent protein binding or lysine acylated histone binding, which makes a lot of sense because these are stimulated with DPA, and that MIAT is regulating these sites that are relevant and important for acetylation. So if we knock down this MIAT upon stimulation of DPA, we do see a significant decrease of H3K27 acetylation compared to the control, suggesting that MIAT does play a role in acetylation of the cells, and that one of the key genes that are dysregulated upon knockdown of MIAT is INO-ADB. And INO-ADB is a nucleosome repositioning or an important factor that plays a sliding of histones as well as a histone addiction in the chromosome. So we are now further investigating whether MIAT can play a role in recruiting INO-AD in the repositioning of histone during the gene transcription activity. So I'll quickly summarize that we've done post-screening to reveal link 816 that modulates MXD1. We also perform molecular phenotyping, which I believe is a very powerful way to understand the function of these unknown genes. And we've shown that LATR1 controls cell cycle through RBP7, and that MIAT could mediate BPA-dependent acetylation in INO-AD regulation as well. So with that, I just want to stop here and acknowledge many people, especially in their initial single-cell atlas study, that many clinicians and collaborators have contributed to this work. And especially I want to acknowledge Chung-Cha Hong, who was a partner in crime in developing the TCRE Atlas paper. And also want to thank Yotaro Shibayama, who also highlighted this localization on RNA work. So with that, I want to stop and thank you for your attention and happy to take any questions.
Thank you for your great lecture. So there are some questions already uploaded. So if you have the audience have any question, upload your question on the website or on the Zoom window. The first question uploaded is this: could it be said that the alternative promoter? You can see the question in the web Zoom window. Could it be said the alternative promoter enhancement related to alternative isoforms or transcript?
Absolutely. So, of course, the alternative promoters can lead to different splicing isoforms, which have of course, a shorter 5' end or 5' ETR. Some have would skip the first exon, and then it would be transcribed from the second exon. So, alternative promoters have a key role in regulatory process of splicing. Of course, splicing can occur also from the 3' end or also in the gene body. Those are a little bit harder to detect using 5' strategy, although there's some trick that we are using 5' strategy, because we do get a lot of exon paintings, meaning there are kind of spurious short reads that get mapped to the exons using 5' strategy, and we're using this noise actually to infer different isoforms. And there's a great work that's being done within the HCA community to do this. But yes, in brief, yes, 5' TSS alternative promoters lead to different splicing, and they are regulated by different transcription factor machinery as well. So there's different motifs that can regulate these isoforms.
Okay, the next question, the paragraph contains many questions. What's the reason for the generating metacells using C cell before the conducting NMF is to reduce the cell count or computing resources? Next question, what is the exact input for NMF? Is it some UMI or other levels of expression from outside cells?
Yeah, I think Metacell for us it helped quite a bit in terms of gaining greater sensitivity. As we know, single cell method still suffers from dropout rates, and we want to take advantage of many cells that we have profiled that exhibit very similar gene expression profiles. So we clustered using metacells and to enrich for especially the non-coding enhancer RNAs because they're also lowly expressed. And that really helped us to infer and define the key regulatory elements in a very specific manner. So that was one of the key reasons for this. So we did summation of all counts in these metacells to ensure that we get sufficient coverage. I don't know if that answered your question. Yeah, I mean, computing resources, I think if you're handling 500,000 cells, it's not much of an issue. I think, so it's for us more of a sensitivity issue that we wanted to have a higher gene detection rate. But key here was that we were able to project it back to single cell by multiplying the module scores to the gene expression. So that really allowed us to visualize these modules as well as these heritability enrichments, these disease traits back to single cell resolution. And we were quite surprised and happy to see that many of them do make a lot of sense in terms of the subtype resolution that we were able to get for these different disease traits.
Okay, the next question is about the ice cream score. She wants to more information about ice cream score. How is it deduced and what's the interpretation? Can you explain briefly?
Yeah, briefly, I would highly recommend you to go to the bioRxiv paper. It's a really good summary of how they are derived. But in a nutshell, it's based on multiplication of matrix tables that are derived from linkage disequilibrium scores to the module scores. And we were able to extrapolate that individual modules could have a score relevant to the disease traits. It's relatively straightforward. And once we were able to derive this matrix multiplication table, we were then able to, again, project it back to the single cell resolution by multiplying to the gene expression level at the single cell resolution as well. So I know it's a very brief description, but if you are interested in more detailed deduction of how we got the ice cream score, I would highly recommend you to go look at the paper. Thank you.
Okay, I will read the next question. He's interested in the functional characterization of RNAs under stress by dividing RNA into origin of the intracellular compartment. Is it possible to determine whether the captured RNA underwent the post-transcriptional modification in case of mRNA?
Yeah, that's a very interesting question. We are also exploring that as well. I think a great way to do this is if you are able to fractionate these cells like we did, but do direct RNA sequencing in nanopore, for example, and to see whether different modifications are found in control versus the perturbed state. I think that could very well be the case because I think the localization, whether it's in the cytoplasm or in the nucleus, have very different function. I could give an example of NORAD, which is a non-coding RNA. In the cytoplasm, plays a role in regulating translation by binding to familial binding proteins. But in the nucleus, it's important in DNA repair mechanism. And these are exactly the same non-coding RNA. And it's reasons to believe that there are structural changes that takes place based on their localization. and this structure very well be determined by modification on the RNA or their nearby binding proteins. So there's a lot to explore of how this same non-coding RNA can exhibit different function is something that's very interesting and relevant.
Okay. There is an uploaded mention that Ju-hyun, who asked about metacell, she uploaded mentioned that she was not able to consider the advantage of enriching low expressed genes by using metacell. So, and she expressed his thanks to your talk. And I think the last question is from Hyunja Kim. Okay, I think it's because he asked this question because it's lunchtime in Korea. So he wanted to buy some lunch food to you. So what's your Korean food? What's your favorite Korean food?
Yeah, I was in Korea a couple of days ago and I had an opportunity to meet Hyunjae. So thank you for the question and thanks for buying me lunch. It's great.
More question uploaded. So I think this would be the last question. What proportion of eRNA in promoter target gene expression is highly correlated? Proportion of eRNA in promoter and target gene expression is highly correlated. I believe that there would be a correlation between them, but it's not always in the case.
Yeah, it's hard to give a precise proportion of which have good correlation, which do not, because it's also very cell type dependent. We do see correlation, which also enhancers are also switching. So I talked about promoter switching, where different cells have different promoters active, but we also observe enhancers switching depending on the cellular context, which makes a lot of sense because these are also driven by, in many cases, transcription factors. And we see that some enhancer promoter pairs are so for example, one enhancer and one promoter is observed in one cell type, but not the other enhancer, but other enhancer is correlating to the same promoter in a different cell type. So it's really hard to kind of give a broad summary of how these correlations are stratified. But we do see that these correlations are highly cell type dependent and they do switch quite dynamically.
Okay, thank you. I think we should close the first session of today's symposium because it's lunchtime and everybody must be hungry. So thank you very much for all the speakers and audience. Thank you.